So, welcome students. Today we are going to learn and see the next experiment in our pharmacology 3 semester 6B pharmacy. Here we are going to learn the evaluation of effect of saline purgatives on frog intestine. So we are going to learn the saline purgative action of our drugs on the frog intestine. So by using this, we are going to see this demonstration of this saline purgative drugs. So what is the today's objective? The evaluation of effect of saline purgatives on frog intestine. So let us see the instruction given here. We have to take the frog and place it on the dissection board, expose the abdominal cavity and carefully place the small intestine. We have to make small intestine into 3 compartments by tying a thread of different colors in such a way that no fluid can come from one compartment to another compartment. So we have to make the intestine or divide intestine in 3 different compartments so that we can tie it tightly so that one fluid from one compartment shall not move from one compartment to another compartment. And by injecting 0.2 ml of each hypotonic solution into first compartment, 0.2 ml of hypertonic solution into second compartments, and the 0.2 ml of isotonic solution into third compartment. So we have to inject the different solutions which are having the different osmotic pressure. So by that way we have to inject in the 3 compartments of the intestine, and after injecting these solutions we have to wait for 20 minutes and the observations are to be recorded after 20 minutes. So let us see how we are going to perform this experiment. So the principle of this experiment is, you can see here, effect. Saline purgatives are the salts which are comprising of the highly charged ions and they do not cross the cell membrane freely. They remain inside the lumen. And retain water through the osmotic process. So they increases the volume inside the bowel and it causes the stretch the colon and produces the normal stimulus for the contraction of the muscle that leads to defecation. And the aim of the present study to examine the effect of saline purgatives in the frog intestine. So this is the principle. So what we have to check our drug does our drug possessing the purgative action or not. So let us see the experiment here. When we are going to see this experiment, we will start here. We have taken the intestine and divided into 3 compartments. Here you can see compartment 1, compartment 2 and compartment 3. We have tied it with the knot of different colors of threads, and we are now going to inject the hypotonic solution into first compartments. The second is the hypertonic solution, which is comprises of 27% magnesium sulfate solution, and third one is we are going to inject the isotonic solution, which is a frog linger in the compartment 3. And let us inspect the observation after 20 minutes. So what we are observing after injecting these solutions, so we have to check the different effects of these different solutions here on the different compartments of the frog intestine. So after 20 minutes we can see that the compartment 1 gets shrink, compartment 2 gets swelled, and compartment 3 get no change. So what we have injected in the compartment 1? That is, we have injected the hypotonic solution here, and so it shrinks so that the content have absorbed the water, while here the cell membrane process the fluid so that it gets shrink. The fluid passes into the tissues so it gets shrink. While in the second compartment, which is the hypertonic solution, which comprises of the magnesium sulfate, here you can see this magnesium sulfate is holding the fluid inside and don't allow the solution to pass through the cell membranes of the intestine, and it remains inside the lumen of the small intestine. So it causes the pressure, and which helps for the defecation of the bowel. So in the third compartment, you have seen no change has been occurred due to the isotonicity solution, and in isotonic solution there is no movement of the ions takes place, so the no osmotic pressure change. Here the osmotic pressure increased, and that causes the defecation of the bowel movement. So here you can see what is the observations we can see. So here in this observations, hypotonic solution causes the fluid to move from lumen into circulation by process the osmosis, thereby shrinks the intestine, that is the compartment 1. While the hypertonic solution, that is saline purgative, moves the fluid from cells into the lumen and swells the intestine. And the third compartment, isotonic solution, did not show any fluid movement across the intestine membrane. So here this kind of observation table you can note on your notebooks. So here you can see the hypotonic solution, your compartment first has the shrinking effect. Hypertonic solution, that is comprising of the 27% of magnesium sulfate, and you can see this magnesium sulfate has the swelling effect, that is the osmotic effect, and the isotonic solution do not show any change in the osmotic pressure. And so we can conclude that the hypertonic solutions, so that is saline purgatives drugs, moves the fluid from cells into the lumen and swells the intestine and shows the purgative effect in the intestine. So that is the conclusion of today's experiment that we have saw. The purgative drugs have the purgative actions and have the defecation, ease the defecation in the bowel. So we can use them whenever the person is suffering with the constipation. We can use them for the relieving the constipation in that patient. So that was the today's experiment. If you have liked this video, you have to share this with your friends who is studying in the pharmacy field. So we will meet you in the next lecture. Take care. Bye bye.